Hi all, Dr. Clark here again, and today we're going to talk about Mendelian genetics, or the theory of heredity. Okay, last time we talked about Mendel in general and kind of the way in which he set up his P experiments. Now we're kind of going to dive into the nuts and bolts on the math and the mathematics around his theory of heredity. Most of you are fairly familiar with simple punnett squares like this one here. And this is a monohybrid cross, meaning that there's just a single character that we're looking at, and that would be the color of the pod in this case. So yellow being recessive and green being dominant. So you can see here this guy is a true breeding green pod, and this is a true breeding yellow pod. Let's just say, for example, this is the female. That would mean that if the female produces two ovas, that both ovas have the possibility to pass on the true breeding yellow pod. And if this pod is the male, and that would mean that the sperm or the pollen from this individual would either have the dominant green pod or the dominant green pod, which would give you this result. 4 offspring, or however many offspring they have, but it would be basically 100% would be non-true breeding dominant for color. Okay? And so we'll look at this in much greater detail as we progress throughout this lecture. All right. So remember last time I was talking about Mendelian theory, and I said that he had 5 main hypotheses, 3 of which are true, and 2 which have been rejected since modern genetics has been formed. And part of that really has to do with Mendel was coming up with his theory of heredity without knowing that there's such thing as DNA, that there was such thing as chromosomes, how chromosomes pair up, how chromosomes move. He didn't know anything about mitosis or meiosis, which we already covered in this course. So it was pretty remarkable to think that Mendel came up mathematically with this theory of heredity without knowing what we were actually inheriting. It's pretty cool to think about. But so since Mendel's time, we now know that genes, or what Mendel was calling characters, are carried on chromosomes, okay? And they have a specific spot on the chromosome which we call a locus or loci. And we've already talked about the general structure of the chromosome, where you have a centromere, and then the small arm is called the P arm, and then the large arm is called the Q arm. But each one is also numbered, so you have a certain region that loci would carry a certain number. Like it would be considered like P4, okay, and that would be considered where that gene might reside. Now that allele, okay, is the variety or the different type of character. So so a different variety of the same character, same gene in this case. So an allele is just a variation of the gene. So you might be dominant for a certain gene. Maybe this is flower color, or pea color, or pea size, or whatever. Might be dominant, and you might be recessive. So in this case, this individual would be a heterozygote. They got a dominant variety from their paternal line, from their dad's line, and they got a recessive variety from mom, or the maternal line. And you could be the same thing. You could get a dominant from paternal and a dominant from maternal, and therefore you're considered a homozygote for a certain genotype. So hopefully that makes sense. That's where the information is coming from, and where you kind of get the letters. So whether you're dealing with big P, little p, big Y, little y, big X, little x, these the notions are really just denoting the chromosome and the gene on that chromosome. So remember, what this represents, what it really is a bunch of A, T, C's, and G's that are listed out in a certain region. That's coding for a specific protein, and that specific amino acid chain. That specific amino acid chain is going to fold in a specific way, and on and on and on. And that's what your genetics is about. So it's much easier for us to just note this as like a big P and a little p than for us to write out 1,000 base pairs and try to do a punnett square with 1,000 base pairs. And so that's really the difference there in Mendelian theory and a punnett square. Now, back to the conversation that we had before when I was trying to get across that just because a trait is more popular in a population doesn't mean it's dominant. Right? Or just because a trait is rare in a population doesn't mean it's recessive. In some cases that's true, but take for example, if we look at dominant traits, like something like this, okay, polydactyl. So the dominant version in humans is to actually have 6 digits on your hands and 6 digits on your toes. We would consider an extra finger or extra toe. That's polydactyl. Okay? That's actually the dominant version. Okay? The recessive version is 5. So, not that there aren't people out there that have 6 digits. There are. Okay? But majority of the population has 5 digits. So it has nothing to do with how often it's represented in nature. What it has to do with is when you take the dominant gene and stick it with the recessive gene, the dominant gene's phenotype is going to show. For example, if white is recessive, okay, and purple is dominant, most pea plants that you ever see in your life are going to have white flowers. Now, wild pea plants will have purple flowers most often. But the only thing that changes here is not how often it's represented or anything like that. It's when you stick the two together, the one that shows up in the F1 generation is dominant. And then you can cross those dominant individuals or those F1 generation individuals, and you'll see the recessive return. That's all it is when it comes to flower color, when it comes to different traits that you see in humans that are recessive or dominant, like the ability to roll your tongue, like the ability to bend your thumb at a 90-degree angle, which sometimes we call a hitchhiker's thumb, whether or not your pinky bends in. There's lots and lots of traits out there that are considered dominant or recessive, single gene traits in humans, and really it has nothing to do with whether they are abundant in a population or not. Okay, so when Mendel devised his Testcross, he needed to understand, well, what is the genotype of the F2 generation? And in order to find out what the genotype of the F2 generation is, he had to devise some way to test it, which he called a Testcross. And the one way that you can do this is by combining any unknown individual, so a dominant individual that you don't know whether it's homozygote dominant or a heterozygote, combine them with a homozygote recessive individual. And this will result in figuring out what that individual is. Because if the unknown is homozygote, then all of the offspring will express that dominant trait, just like a true breeding combination. True breeding white, true breeding purple. If that comes out and you have all purple plants, and then you can just take those purple plants and you mix it with the white plant again, and you get the heterozygote cross with some of them, okay? Which would mean that about half, okay, or a 1-to-1 ratio of the offspring will express that recessive trait. Okay, hopefully that makes sense. We're going to look at a little diagram. So let's say you don't know. You know that this is the dominant phenotype, but you don't know whether it's homozygote dominant or heterozygote. So you can just cross it with a homozygote recessive individual. If all the offspring show the dominant trait, then you know the original parent showing the dominant trait was homozygote. If the offspring show a 50-50 or close to it, then you know the original parent was a heterozygote. And so this is just an easy test that Mendel designed to find out what portion of the F1 generation were heterozygotes versus homozygote dominant. Okay, now for a little more complexity. So those were monohybrid crosses. One trait, okay? One trait from each parent. So purple, white. Okay? Now we're going to look at dihybrid crosses. A little more complex, but nonetheless mathematically fairly easy to figure out. Okay? So now let's say we're interested in crossing a round yellow seed with a wrinkled green seed. Okay? And round and yellow is dominant and wrinkled and green is recessive. So here you can see how that's set up. Okay? Each one would be getting a chromosome from their parent strand, so paternal, maternal, so you have to have two letters. Big R would indicate that it was round. Big Y would indicate that it was yellow. Okay. Little r indicates that it's wrinkled. Little y indicates that it's green. Okay. And through meiosis, remember the process of meiosis, that you're going to inherit something. The sperm, okay, is going to have one possible character, and the egg is going to have another possible character. In this case, the possibilities are the exact same. Right? You have big R for round, and big Y for yellow, little r for wrinkled, and little y for green. That's your only combinations through meiosis. Fertilization occurs and you get a heterozygote. This individual is going to be round and yellow, but it's going to be a heterozygote. It's going to carry the recessive traits. So now we're interested in, well, what's the possible offspring from this individual? Okay. They could be big R, big Y, show dominant for both. Big R, little y, so show dominant for shape, but recessive for color. Little r, big Y, so recessive for shape, dominant, or sorry, yes, recessive for shape. Holy moly. Big R, or little r, big Y. Yeah, so recessive for shape, dominant for color. Wow, that was a struggle for me. Okay, and then little r, little y is recessive for both. So those are the possible gametes. So if this is going to be a female plant, these are the possible eggs. If this is going to be a male plant, these are the possible sperm or pollen. Then you can cross these two. So you can cross this generation with itself, and this is what you get. When you cross during a dihybrid cross, when you cross two heterozygotes, you will get a ratio of 9:3 to 3:1. Okay? And what this amounts to is about 56% or so will be dominant for both characters. 18% will be dominant for one, and 18% will be dominant for the other, and then about 6% will be recessive for both characters. So it's not that you would actually produce 16 offspring necessarily. Maybe some organisms that you would do this with would, but it would just give you the probability of, if you're producing one offspring, you would have a 56% chance that it would be yellow and round. Right? And that's how Mendelian genetics, the purpose of Mendelian genetics, or the theory of heredity, really wasn't about, you have to produce 16 offspring, but really it was, what if you produce one offspring? What are the chances that that offspring would be dominant for a single character and recessive for the other? 18%. So that really brings us to what we call Mendelian law. Okay? Now, it's not necessarily a law per se. It's a mathematical law in a case, because you can prove it, because it's there's an equation for it. But again, it's based on Mendel's hypotheses and his results from his research, and you kind of get two basic laws. And so the first one is what we call the law of segregation, and that is that alleles of separate, or traits, are separated from each other during the formation of gametes. We already know this. Okay? And so half the gametes will carry one copy, and half the gametes will carry the other copy. So this goes directly to meiosis. And if you remember, meiosis is a reduction division. You're either getting chromosome number 1 from your mother's line or chromosome number 1 from your father's line. That's where Mendel's first law of segregation comes in. Remember, when he came up with this, he did not know that alleles, he did not know that genes are carried on chromosomes. So really all he's doing is telling you how chromosomes separate without knowing anything about chromosomes. The second law that Mendel came up with, okay, is the law of independent assortment, which should sound a little bit familiar to you because that happens in metaphase 1 of meiosis. And so that means that genes are located on different chromosomes, and you inherit chromosomes independent of one another. This is true. We know that. You only inherit either dad's chromosome number 1 or mom's chromosome number 1, which means that you would inherit the genetic material on that chromosome independent of each other. And so that's where Mendel's laws come into play. It really has to do with chromosomal inheritance. But remember that Mendel did not know about chromosomes. So this is pretty interesting stuff, because he's talking about genes or characteristics and concluding that this is the way it must be inherited, but he didn't know there was such thing as chromosomes. Okay? And we'll come back to this and we'll figure out, I'll talk to you about a couple individuals, Sutton and Morgan, that Thomas Morgan and might be William Sutton, don't quote me on that, that came up with chromosomal theories that are basically identical to this, and that's where Mendel was getting at with Mendelian law. All right, so how are genes influencing traits? Remember back a long time ago, I talked about something called the central dogma theory, okay? That DNA is changed into RNA, and then we call that transcribed. Okay? Then RNA is used as a blueprint to form an amino acid chain or a protein. Okay? So genes are just DNA, and DNA is translated into RNA. That's the language that ribosomes can speak, per se. They understand it, okay? Or at least transfer RNA understands it. Okay? And then amino acid sequence can be built off of the RNA strand. Okay? So this is DNA is transcribed, RNA is translated. Okay? That's how genes influence traits. Okay? So again, the RNA is translated into polypeptide chains, amino acids. And remember, just the simplest form of a protein is really just amino acids linked together. How they are linked together determines how that protein is going to fold. Okay? And we've already talked about most of this because we talked about proteins and we talked about enzymes, we talked about amino acids, and talked about DNA. So this is just reiterating, if you change the DNA, you change the RNA, which then changes the amino acids laid down, and that's how genes influence traits. Okay? Because all a trait is, all a expression of DNA is your phenotype, and that's based on the polypeptide amino acid chain, which is based on the RNA sequence, which is based on the DNA sequence. So again, anytime you change the DNA, it can affect the order of amino acids laid down, which can alter the arrangement of the protein, so it folds in a different form, reflects a different light, has a different overall function. All these kind of things can occur, which gives it a different phenotype. Okay, so here you can see what we're talking about, okay, in diagram. So remember that chromosomes are housed inside the nucleus of eukaryotes. Now, we're not just talking about eukaryotes here. Prokaryotes have chromosomes also and have the same function. Okay? So again, we're just showing you the human genome. Okay? We have about 3 billion base pairs and a little over 25,000 genes on 23 chromosomes. Okay? And so chromosomes housed inside the nucleus. Most chromosomes have somewhere around 1,000 genes on them. Okay? Some a few more, and some a few less, to range around 25,000 genes, somewhere in that range. Okay? Well, one of those genes, we're talking about hemoglobin here in this diagram. One of those genes is we call the HB gene, or the hemoglobin gene. Okay? That's going to be copied by RNA. So you make an RNA copy of it. That can then leave the nucleus. Okay? That RNA copy will then encounter ribosomes, and through transfer RNA and some other things, which we'll get to, the amino acid sequence that corresponds to this RNA code will be laid down. Then that amino acid sequence is allowed to come free of the ribosome and things like that, and will start to fold. And it folds in a certain form based on the amino acid sequence. And that gives you a HB protein, which we call hemoglobin protein, which we find inside red blood cells and allows for us to carry oxygen and carbon dioxide to oxygenate cells and rid our body of carbon dioxide gas. Pretty simple. Now, you make one tiny little change, and you change the RNA, you change the amino acid sequence, you change how the protein is folded, and you can go from having a normal hemoglobin protein to having what we call sickle cell anemia, which means this protein is different. I don't want to say it's worse or better, and we'll talk about the evolution around this. It's not as good at picking up oxygen. So individuals with sickle cell cannot live in environments that aren't rich in oxygen, but nonetheless, it's still there. And the individual can still live and pass on that mutation or that change in the hemoglobin gene. Okay? We'll come back to this. We'll talk a lot more about the evolution of having these mutations and why we still see some of these mutations in humans way past their prevalence and maybe even past their usefulness, but we still see it. Alright, next time we are going to talk about the exceptions to Mendelian genetics, which there are a ton of them. Okay? So stay tuned, and I'm going to talk about where kind of Mendel's research with peas, it wasn't good enough to tell us everything that we needed to know about genetics, and it just told us a very simple form of genetics. Now we're going to go a lot more complex and see what happens with different types of genetics when it comes to phenotypes of humans and all other organisms, actually. Next time.